Okay. Welcome everyone to BIMAC Colloquium. I have the great pleasure of introducing today's BIMAC speaker, Professor Sam Isaacson, who is the professor of mathematics and statistics at the Boston University. Professor Isaacson is a highly accomplished mathematician and researcher in mathematical biology. His research focusing on developing the numerical analysis and method for studying problem in molecular biology. Recently, his research has focusing on the stochastic and deterministic reaction diffusion method, appropriate for modeling biological system with the multiple scale. I'm confident that you will find his talk today to be both informative and engaging. So please join me in welcoming Professor Sam Isaacson.
Thank you. Should I start?
Sure.
Okay, great. Yeah, well, thank you for inviting me. It's great to have a chance to give a talk to everyone there. Many of you, I guess, or maybe all of you on the other side of the world, and maybe I'll see some of you next summer at SMB.
Yes.
So I thought I would tell you about a project with my collaborator, Omer Dushik's lab at Oxford on trying to develop models. Originally, we were looking at, and we're coming back to now, to try to model how T cell receptors interact with ligands for them. So these are receptors of on the surface of immune cells. But today we're going to focus on kind of a detour we got taken into, which was interactions between antibodies and antigens, specifically antibodies to COVID from patients and RBD, the receptor binding domain of COVID. Please feel free to ask questions as I go, because I don't know everyone's background. I don't know if I put too much biology and should have been more mathematical or vice versa. I tried to put a bit of both. So a lot of people have worked on this. I'll try to point out their contributions, but Anna really did is a great graduate student in Omer's lab who really drove a lot of the analysis and kind of hypothesizing and applying both the mathematical methods and simulation approaches we developed to the data that was being generated in Omer's group. Dan is a postdoc that did some MD modeling, molecular dynamics modeling. Dan Wilson was a postdoc with me who worked on some of the methodology and is now at the Davenport lab in UNSW. And Mikhail's the one that did all the experiments, actually ran the experiments that I'm going to show you today. And if I can advance my slide. Okay. So I've been working with Omer's lab for quite a while on a variety of problems, generally around how one could try to estimate binding kinetics in mathematical models and biophysical properties of various membrane-bound systems. So we originally looked at what's called tethered enzymatic reactions, where you have a phosphatase like SHIP1 that can bind to the cytosolic tail of, so imagine you have a protein in the membrane of a cell facing inward and it has a tail, and then these SHIP1s can come and bind. We were trying to look at that and model that binding process and figure out rates for it and such. Then kind of after that work, we got interested in, Omer's worked on this for a long time, in whether we could kind of adapt these methods to try to study and estimate parameters in how T cell receptors on the surface of T cells can bind to antigen or peptide major histocompatibility complexes exposed on the surfaces of antigen presenting cells. And we kind of started working on that, and then COVID hit, and then Omer kind of pivoted to do some COVID stuff for a while. And that brings us to what I'm going to talk about today, where I'm going to talk about looking at antibody and modeling and doing parameter inference on antibody antigen binding. And so for today's talk, we're going to be looking at the antigen that we'll talk about will be the receptor binding domain of COVID. And the antibodies, like I said earlier, will be patient-derived monoclonal antibodies. And so this is kind of a picture here of a single antibody bound to two RBDs. And so one of the things, if you don't know antibody biology, is they have two binding sites, and we'll think of them as independent and identical. So here the FD11A is in the middle. So that's the bivalent antibody, the bivalent indicating that it has two binding sites. And then here you see the RBD bound to those sites. And so we're interested in trying to understand the kinetics of that binding process, and more importantly, to try to be able to develop methods to predict how good is an antibody going to be at neutralizing or sequestering these RBDs and ultimately preventing COVID from infecting cells. That's kind of a higher level goal. So kind of long-term goals, like I said, are understanding and predicting antibody function. An important one that you'll see a bit of today is how do the kind of parameters that come out of kind of biophysical and kinetic models of these processes relate or do at predicting the ability of an antibody to neutralize the antigen, that is to shut down the RBD and prevent it from binding to a cell. We're trying to develop a mathematical modeling and computational approach that would allow Omer's lab to take panels of many antibodies, tens, hundreds of antibodies, and run them against an antigen and develop parameterizations of the binding kinetics so that they can then try to predict which antibodies are better at binding to it than others. And what we've kind of switched back to now, which I'm not going to talk about today, is applying some similar techniques to what I'm talking about today to look at T cell receptor function. So, okay, so in today's talk, the models are all going to be built to model an experiment and to then fit and infer parameters from the experiment. The experiment that we're going to be modeling is what's called the surface plasmon residence, or SPR assay. I will not pretend that I really understand the physics of how SPR works. So if you ask me detailed questions, I won't be able to answer them. But in my naive worldview, you have a sensor surface here, and you are able to shine a light on and measure changes in the refractive index of the light as the amount of stuff on the surface changes. And so you get kind of a single signal across the whole device, and I'll show you example data in a minute, given in arbitrary response units. But what's useful about this signal is it's directly proportional to the change in mass near the surface. So basically, when the signal you get goes up, we assume that more stuff is binding at the surface in a proportional way, and when it goes down, it's unbinding. Okay, so the signal allows us to measure essentially binding in this case of I actually don't know what's being. I guess these are probably antibodies binding to antigens, or maybe these are receptors binding to ligands in this picture. But that's kind of our naive picture or naive setup for how I'm going to think about it. So to show that a little more concretely and maybe a little more mathematically, let's look at kind of a standard model for how one might model these experiments and try to predict what's going on and then be able to fit the model to actual experimental data. So this is what's called a monovalent SPR experiment. And the standard picture is I have antibodies that I bind or attach to the surface, and then I flow on top of them antigen. And if I assume that the binding to each arm is an independent event, then essentially if I have N antibodies, I have 2 N binding sites, one for each arm of the antibody. And in the simplest case, you assume this is well mixed. And so there's just a rate that an antigen can bind to an arm of an antibody, and there's a rate it can come off when it's bound. And hopefully that makes sense to everybody. I'll write equations in a minute. One thing I want to point out, because this picture is a little misleading, is that in real life, the picture here makes it look 2D, but actually these antibodies that are being bound to that are on the surface are actually tethered in 3D. So actually you have a 3D region near the surface of the chip where all these antibodies are tethered to polymers in kind of a random way. Okay, so the models that we do will actually look at things in 3D, but the kind of pictures that I show you as illustrations are going to be 2D just because it's easier to draw. So just keep that in mind as I continue going. So what you see here is a kind of typical set of experimental data. So if we look at one of these curves, they're flowing over the antigen at a fixed concentration. Each curve is a different concentration. So this is higher concentrations of antigen.
So, Sam, sorry. So you mean the 3D means there is a plane where the antibodies are located at randomly on the plane?
There's a height above the plane where they can be randomly distributed in that volume.
Oh, the antibody can, in my understanding.
So instead of them being set up in 2D like this, they're actually set up in 3D. This is looking on the side. That's looking on a side on a cross section. So this is the z-axis. This would be like x or y, and then there'd be another. Does that make sense?
So that means this because you said it's some surface plasma resonance. So I thought that there should be some surface.
Yes, but they're able to actually measure the binding in a neighborhood of this. So the change is actually be able to be detected from a neighborhood. So they actually put them in a small region. Here it says 100 nanometers in this example. It would be the z would be kind of the z distance.
Okay.
So it actually is a volume that they're distributed in. They're not just lined up. You can set up surface plasmon resonance assays where they're just lined up on the surface, but I'm told that they're a lot more difficult, and they're much harder to actually figure out like what the concentration of the antibody on this in the region is if you do a 2D setup versus a 3D setup, for example. So, okay. So what this plot here shows is some set of experimental curves. Each one of these is for an increasing amount of the antigen, that is the receptor binding domain of COVID. And you see kind of two phases. So initially they start the antigen flowing over and you see binding. So the amount of antigen bound to the surface increases.
Okay.
And then at a certain time, they turn off and they wash out all the free antigen. And so you just see unbinding as stuff leaves the surface. Okay. And so the reason they do these two things of doing the experiment with many different antigen concentrations and then two phases to the experiment is to give the curves more features for when you do fitting. Okay. So it makes it easier to do parameter estimation if the curves have more features in them. That's the main reason they kind of do it like this. Okay. So that's kind of a typical set of experimental data. So what would be kind of the mathematical model? So the simplest model that one could build for this, and we're going to get more complicated as we go, would be to assume a simple reaction of this form where this constant concentration of antigen A, which I'll call AG, or AG, I'll often write it, can bind to unbound antibody binding sites. So there's twice as many binding sites as antibodies. And then they form a complex with the antigen attached to the arm of the antibody, which is C. And so you can write down kind of the simplest model. If we assume that the amount of antigen in the solution is fixed, then you just get a simple set of linear ODEs, which probably everyone, or hopefully everyone, knows how to solve. And so you can solve them analytically, and you get a solution of this form where here we've taken into account that at a time TS, we switch off the flow. So at time TS, we set the amount of antigen to be 0 just to model that this is gone and stuff just unbinds.
Sam, I have a question.
Yeah.
So how experimentally they can keep the constant concentration of free antigens?
Sure. So, I mean, you're asking basically about a detail of how the SPR experiment works. So, so basically Omer has been doing these experiments for a long time, and they have ways that they try to set the flow rates and the concentrations that they feel confident that you can model it as a well-mixed system in that regard. However, if you look in the literature, you'll see an infinite number of papers trying to model mass transport effects. And so, and the first thing that someone who's an expert in this will always probably bring up is, well, how do you know mass, transport's not an issue? And Omer's answer will be, well, look at all the experimental validation we'll show you later in the talk to show that we're making good predictions consistent with other independent experiments.
Gotcha. Thank you.
Yeah. So that is definitely an issue with SPR that you have to be worried about is, are you really have a well-mixed system? But let's assume that we do. Okay. So we have an analytic simple formula for the amount of bound antigen at the surface at a given time. How do we relate this to the measured response, which has arbitrary units? And so the general assumption is just that the two are proportional. So the measured SPR experimental response is proportional to the solution of the ODE model for the amount of stuff bound at the surface. And so we end up having three parameters in this very simple classical monovalent model: the on and off rates for binding and unbinding, and this constant of proportionality. Okay. So we have three parameters to fit to that set of curves. And so typically what you do then is you run with a bunch of different antigen concentrations, you get a bunch of curves, and then you do a fit of these parameters to all the curves simultaneously. So you don't fit a
Yeah.
set of parameters for each curve, but you fit across all the curves.
Yes.
And so this here is just showing an example of an antibody to COVID called FD11A, and showing you the fit that we get out in that case. And you can see the fits are quite good, right? They look pretty good overall. So that's kind of the classical picture, kind of the standard setup that people started with, but there's some limitations to that approach. So one is it only provides what are called monovalent rates. That is this kind of bulk rate that you bind to a free arm from the solution. But if you think about the picture of how antibodies bind to antigens on the surface of a virus, it's a little different. So imagine this is my viral surface. These are my antigen receptor, the SARS-CoV receptor binding domain of spike protein. And these are some antibody to it. So the typical process will be my antibody will come in and one of its arms will bind. But now once this other one is tethered here, the other one can kind of flop around and try to bind to another antigen.
Yes.
Okay. So it's not clear that in this kind of picture of a more biophysical representation of the model, that the second binding rate should really be the same as the bulk binding rate from the fluid. But another really important issue is that you can't bind to all of these antigen across the surface. You can only bind to those that are close enough. So there's kind of another parameter that comes into play that we might want to account for, and we'll get back to this later, which we call the reach, which basically describes if an antibody is bound to one antigen, how far away can another antigen be for it to be able to bind to the second antigen?
I see.
That is, how far away could this guy be so that this can bind to it? Okay.
I see.
And
So
that's
you
really
mean that the second binding cannot target the old antigen, right? So only the locally they can bind.
Only those that are close enough to be within reach, to be able to actually encounter this free arm.
So
that's right.
So that means first binding and second binding are qualitatively different. So first binding target everything, but second binding is really narrow zone. Wow.
That's right. It's a more microscopic model we'll end up having for how the second binding occurs.
Wow, the problem become more difficult.
So yes. Okay. So that's one issue, is that the monovalent experiment doesn't look so much like what happens in vivo in cells. Another issue is more experimental, which is that the way the monovalent experiment was done, we put the antibodies on the surface and we flow over the antigen. And this is the hardest part of the experiment, is getting things attached to the surface. And what the experimentalists would like to do is pick one antigen and test many antibodies against it to compare how well they do at binding to it. You know, to try to say, I have a panel of 80 antibodies, which are the best candidates for using for treatment. So they kind of would like to be able to do the reverse experiment where you send over the antibodies and the antigen are fixed. And so you only have to attach one thing to the surface one time, and then you change what you flow over. So that's another issue. And then the final issue is that these K-on and K-offs you get turn out to not really correlate very well with how good an antibody is at shutting down spike protein, at preventing spike protein from infecting cells. So you can get these on and off rates, but they don't seem to correlate super well with neutralization ability. And so another question is, if we do a more detailed, physically accurate model, could we better predict neutralization ability of antibodies? So what I'm going to focus on for the rest of the talk is what we'll call a bivalent model that's more like the picture I just showed you. I'll show you how we built the model, how the experiment works, how we kind of built a computational workflow to combine them all. And then I'll show you how well it does kind of at predicting the ability of the antibodies to neutralize just from the measured parameters. So kind of the simplest bivalent assay you might think of as kind of the analogous one to the monovalent, but flipping what's free and what's bound. So now the antigen are on the surface, again in 3D, not 2D. This is just representative. And the antibodies are flowed over. Well, now you'd have a two-step process where the antibody binds, and then there's some rate that the second arm on the antibody can bind. Okay. And so kind of, and these models have been looked at before in the literature. So now we would have three states we keep track of: the amount of unbound antigen, the amount of antibody antigen complexes with one bound site, and then the amount of antibody antigen complexes where the antibody is doubly bound. Okay. And so we can write a simple reaction model for that. You know, a free antigen gets singly bound by an antibody, and a free antigen and a singly bound antibody-antigen complex form a doubly bound antibody, and then write the corresponding ODE model, which we just get this. So it's still a very simple model. It's easy to fit it to data. Now what we assume is that the SPR measured response is going to be proportional to the amount of antibody that's bound at the surface, because we're having the antibodies come in and bind. So
Sam, I'm sorry. Can you show the previous diagram one more time? So
yeah,
this
picture.
On the next one. So B is a single bound one, right?
Yes.
Okay, and then if
hopefully I just typed these up the other day, so hopefully I didn't make a typo in typing them
so
up.
I'm a little bit confused. So maybe
this is the concentration of antibody. So it's like since the antibody is well mixed, it's like a first order rate that free antigen turns into singly bound antigen.
Okay. And
then a singly bound antigen with antibody complex, the antibody can come off with some rate.
Yes.
There's a separate rate that a free antigen and a singly bound complex can form a doubly bound complex.
But
and then
but we need a local.
There's no space. This is non-spatial, so we're not.
I see. I see. Just typical ODE approach. Okay.
This is the analogous bivalent ODE model.
Okay.
First, I want to convince you that this model is no good.
Then I see. Okay, makes sense. So because it didn't make sense to me, so that's why.
No. Sorry. So this is kind of the next simplest thing one might try if you flip the experiment. This is the analogous ODE model.
I see. So you assume that even second binding is global, not the local.
That's right. It's still global.
Okay.
That's right. And we'll see that that's a problem.
Okay, gotcha. Thank you.
Okay. So like I said, now the SPR data, the response they measure experimentally, will be assumed to be proportional to the total amount of antibody bound on the surface, because that's what's changing. And now we have 4 parameters to fit because there's the second binding rate. Okay. And so you can go and you can fit it. And here's what you see if we do the same kind of setup. The only difference here now is each curve is different antibody concentrations for the same antibody, the FD11A, and then we fit collectively to all the curves. And you see this is a way worse fit. If you look at like this top one, there's a huge error in the fit that you get. Okay. And moreover, if you look at the parameter estimates, so this is the parameter estimates for the monovalent, the first model, for the FD11A when the experiment was run 4 times, and then this is for the bivalent ODE model where it was run 6 times. You can see you have terrible agreement in the off rate.
I see.
The on rates, maybe you could argue there's a little bit of agreement. I don't know, but the off rates are terrible, and therefore the dissociation constant is not very good. So you would expect that you should recover the on and off rates of the monovalent model in the bivalent model, and you're not recovering them, it looks like. And so it seems like this model just doesn't work very well. And of course, when you look at it, since I gave you the picture, one natural thing to think is, well, one of the problems is that, as you kind of mentioned, you're letting the antibody that's bound to one antigen bind to any other antigen instead of just the ones that are close. Okay. And so that was one of the things that we thought when we looked at those models long ago, would be one potential way to improve the model, would be to put in the notion that you can only bind to antigen that are nearby you. And so we want this reaction to be spatial. A and B can only react if they're sufficiently close. Okay. Also, though, we tried doing various types of moment closure models and some kind of mean field models, and they didn't seem to work very well. And one of the things we thought might be going on is that it's hard in those models to capture the fact that if you only have a small number of antigen near a given antigen, then you can get depletion. You can get that all the possible antigen that an antibody bound here could reach are already bound to someone else, and you lose your binding partner. And that's something you can capture in a stochastic model quite nicely. That's harder to capture in kind of a PDE type model or a spatial deterministic model. So because of those 2 effects, we decided to build a stochastic particle model of this experiment and try to use that to kind of fit to the SPR responses.
Nice.
And so now the picture kind of looks like this. your antibodies come on and bind, and now they still can bind with some second rate, but they can only bind to antigen that are close enough, i.e. they're within reach, and we'll call it reach epsilon, okay? So there'll be some probability per time that a singly bound antibody can bind a nearby antigen if they're within epsilon, K on B being that probability per time. And what we'll assume in terms of relating the model to the experiments is that now the SPR experiment response is proportional to the average number of antibodies bound on the surface across our stochastic simulations. Okay. So just to be a little more mathematical, since you said that a lot of the people in the audience would be mathematicians, I thought I would actually write out the equations just for those that know these kinds of models. So
Sam, I'm sorry, one more question. So sorry. So when you calculate the response, so B is a single bound one and C is a double bound one, right?
So adding that up tells you the total number bound near the surface, right?
Yeah. So I'm just curious if double bound, don't we have a double responses or not?
No, because we're thinking of it as the additional mass of antibodies near the surface. So whether they're singly or doubly bound, we're not thinking of them as contributing different amounts of mass.
Okay, I see.
Yeah,
okay.
That's right.
Okay.
Yeah. Okay. So now the model is going to be the following. We're going to, in 3D, we'll just do a cube for simplicity. Put down a random number of, a random set of antigen particles, just uniformly placing them. I think we do about 1,000 of them normally. I'll come back to that point. We assume then that that throughout the whole volume, there's a constant concentration of antibodies. And then we keep track of a collection of stochastic jump processes of this form. So one set of them tells us gives us the state of the ith antigen. So this is 1 if the ith antigen is free at time t and 0 if it's not free. One of them is 1 if the jth antigen has an antibody singly bound to it at time t. And then this one is 1 if there's an antibody bound to antigen I and j at time t, okay? And so these collection of stochastic processes will now describe the state of our system. And I'll call the positions that we put the antigens in space by xi. And if the math confuses you, I'll show a movie in a couple minutes. And so you'll be able to visualize what's going on. Hopefully that will help. So the reaction model becomes this: an unbound antigen, the ith unbound antigen can just convert into being singly bound like a first order reaction. If the unbound antigen at location I and a singly bound antigen at location J are within epsilon of each other, then they can react with some rate K on B to form a complex where the antibody binds the two of them. And then the antibody can come off. If it's bound to two antigen, it can pop off one of the two with some rate. Okay.
So here I have one more question. So AI to the BI is 2 times KON. The double comes from because it can have 2 chance where you multiply 2 to the,
because you have 2. That's right. So that's just, I mean, so to be honest, in the actual simulations, this is just a parameter for me. But to compare it to the monovalent model, this is how we write it, because if you write it like this should be the same rate that you estimate in the monovalent model.
Okay, gotcha.
And this should be the same K off that you estimate.
Gotcha.
Yeah. Okay. So this is just kind of a decomposition to allow us to compare the two models more easily. So you could make this one effective parameter, and that's actually what we do in the simulations. So, whoops. Okay. Any other questions?
No, perfect. Thank you.
Okay. So just a little more then to describe this mathematically. So then let's let I hat be our set of possible antigen pairs. So here's one antigen, all the possible locations if there's n of them. And then if we look at j going from I plus 1 to n, that gives us all distinct pairs of 2 antigens. And so our model is this collection of jump processes. And the way that we'll write down how they evolve is by in the following way. So we'll have a whole bunch of independent unit rate Poisson processes, one for each antigen for the first reaction, one for each antigen for the second reaction, one for each pair of antigens for the third, and one for each pair of antigens for the fourth. And so then we can write down the number of occurrences of each reaction up to time t like this. So for example, for AI goes to VI, then it's just the this particular unit rate Poisson process evaluated on the integrated intensity of the process, gives us the total rate or the total number of occurrences of the reaction up to time t. This is the total number of occurrences of VI go to AI, and so on and so forth. And so having defined these, we can actually write out how the processes evolve in time. They evolve like this. And this is just the time change representation of Kurtz, just written a little more compactly, because if I try to write all the ends in, I can't fit it on one slide. So it's kind of the standard way that you can kind of write out the dynamics of the jump processes from a process representation or point of view. Okay, so once you've kind of set up your model like that, in practice, the rest of what we do is via simulation. We don't do much analysis on these processes. So you can simulate them exactly using the stochastic simulation algorithm, i.e. Gillespie method or kinetic Monte Carlo or Dubs method or whatever your favorite name for it is. We use this Gibson-Bruck method, which if you study these things, you know that the Gibson-Bruck method is actually for chemical systems usually one of the slowest methods. But in this case, it works pretty well because we don't have larger population numbers. And then basically the way we estimate or model the response, the SPR response, is we just sum up all the singly bound antibody states and all the doubly bound antibody states to get the total number of antibodies bound at the surface. We take their average, and we assume that that's proportional to the SPR response again. Here we do a normalization by the number of antigen, but that's arbitrary. We could have absorbed that into that unknown constant. So before I talk about fitting these, let me just show you, if you're confused at all, what one of these simulations actually looks like. And this is just a toy illustrative simulation. The parameters are not realistic. The length scales are not realistic. It's just to kind of show you what a simulation looks like. So here you see in black are unbound antigen. Blue is when an antibody is bound to them. And then these lines linking them up indicate that the antibody is linking those two. It's bound to link those two. So this is what a typical simulation looks like. So nothing is moving. We're assuming everything is fixed in space. It's tethered into the matrix. And it could probably is sloshing around a little in real life, but we're going to ignore that and just treat it as static. But that kind of shows kind of just a picture of what one of these simulations actually looks like.
I see.
Okay. Great. So first thing, before I go into trying to do parameter estimation with this, is how does this reach parameter epsilon, just to get a little intuition, how does it kind of impact the dynamics of the binding process? And so here's 3 different cases. I think we did these ones as 2D simulations, but everything else is 3D. This is just illustrative. So here you see when the distance that a bound antibody to an antigen can interact with another antigen is 2. So the antigens have to be within 2 nanometers for them to react. And you see basically none of them are that close. Here's when it's 10. And so you see in red that there's some pairs of antigens where a disk of radius 10 about them overlaps, or sorry, of radius 5 about them overlaps. And therefore they're within 10 nanometers of each other. These ones are not close enough to another. And then here when it's 30, almost all of them overlap. So almost all of them have neighbors where an antibody can bind to 2. And so you see these kind of different responses. And the main thing I want to point out is that where you see a lot of the changes in the second phase. Basically, when you have a larger reach, you get more bivalent binding, more cases where an antibody is bound to 2 antigen, and therefore it takes longer for it to unbind from the surface. And so that's kind of, if you look and compare these, you see it stays on the surface longer. It stays bound longer. Okay? So that's just hopefully a little intuition on what should happen as we change the reach. Okay, so how do we actually fit the model to data? So we've got 5 parameters. We added 1 more, the reach. We've got the on and off rates that should match the monovalent model, the second binding rate, this reach, and then this constant of proportionality. And we typically fit using this exponential natural evolution optimizer XNES. So it's a global optimizer. It seems to work pretty well. We typically do like 100 or 50 fits and take the one that's the best because it's a stochastic optimizer, so it gives you different answers each time. And what you can see here are 3 different sets of fits. This is fitting the ODE-based model that I started with, the monovalent model. So this was the first model I showed you. This is the second model, which is really bad, that I showed you. Notice this one, we're varying antigen. We're changing the amount of antigen we flow over. Here, we're changing the amount of antibody we flow over because it's the flipped experiment. And then this is our new model where we're flowing over antibodies still. And you can see it's not completely perfect. There is a little bit of discrepancy at the tail, but it's much, much better than the ODE-based model. It really gives much better fits, okay? So that's kind of the basic setup, except there's one little hiccup in all this, which is that, as probably many of you know, running stochastic simulations is very computationally expensive. So to calculate the average response from the simulations, we have to run a lot of simulations and average them and try to make sure that our sampling error is low enough that that's not impacting the fitting process. And we typically try to get the sampling error below about 1% as much as possible. To make this fitting easier. Of course, the biological data itself is not, super highly accurate, but we want to make sure that this is not a huge contributor. But even doing that, it becomes very expensive. And this is a challenge or annoying to the experimentalists because they want to get their experiment data and immediately go and fit and see what the parameters are. They don't want to wait half a day and have to run on a cluster or something and then do data analysis. So what we did to speed up the workflow, this is kind of more of our numerical approach. And I'm always, Jay, you heard me say this at SIAM. I'm always embarrassed to tell this to mathematicians because I feel like we did the simplest thing you could think of, and yet it works really well. So we haven't done anything more sophisticated. So what we noticed was the postdoc who was working with me kept rerunning the same simulations over and over and over. And so at a certain point, I said to him, why don't you start saving your simulation data? And so you don't have to keep rerunning them. And then we started thinking, I said, well, actually, if we're saving tons of simulations over parameter space, couldn't we interpolate them to approximate what the simulation result would be at parameters that we haven't simulated?
Yeah.
And it turns out that if you do that, it works. And it's the easiest thing you could think to do. So what we basically do is we make what's called a surrogate model by taking our three rate parameters and our reach parameter and tabulating the rate parameters over a region of log space, the reach over linear space, and then saving 601 time points of the average response in the model. And we just build a giant linear interpolation table. So for each of the tabulated points, we have a curve. If you give me a set of parameters that I haven't tabulated, I just interpolate the nearest ones that I have simulated. So it's the simplest thing you could think to do. And that allows us to make a predicted response. But it seems to work pretty well, as I'll show you. So the nice thing with this approach is we can go and I can put it on a cluster and run it for 4 hours and run on 500 CPUs, save all this data, and then I can just give this to the experimentalists, and now they can go and they can fit you using the codes, and they don't have to wait. It's instantaneous for them. So there's one little thing I glossed over in talking about the surrogate there, which is that the surrogate also, if you think about it, the simulation results are going to change as you change the concentration of antigen in the system, as you change the concentration of antigen that you put down, i.e. the number of particles per unit volume that you put down on the surface. But we didn't want to have another parameter in the surrogate. So instead, in our surrogate, we just picked a fixed concentration with 1,000 particles. But then we have a little way that we came up with to map fittings from the surrogate with this incorrect antigen concentration to what you should get with the correct antigen concentration. So the basic idea is, suppose we fit our surrogate with this incorrect surrogate antigen concentration, and we get some reach that we'll call the surrogate's reach out. In the experiment, we know what the real concentration is. And what we're going to enforce is that all that really matters in terms of the spatial aspect, basically, is that the only place space comes into play is essentially in setting for a given antigen, what are kind of the number of other antigens that are close to it. That's really the only place that space comes into play. And so what we essentially enforce is that the average number of antigen that are within reach of a given antigen in the surrogate, which is the volume of the region where things can react times the concentration of antigen in the surrogate, should equal the average number of antigen that are within reach physically in the real experiment. So we know this parameter, we know this parameter, we know this parameter. This allows us to solve for the reach, the physical reach, and back out without having to, in the surrogate model, actually tabulate over antigen concentrations too. Okay. Yeah, so that's kind of the basics of the fitting process. So we kind of put a package together for this that does the forward simulations, that can build the surrogate, let you, build it out on a cluster, and that lets you take SPR data that's been processed and cleaned and do the parameter estimation, and then spits out the biophysical parameters. And so this is kind of where the experimentalists use it, is we run this part for them, we give it to them, and they can just do this part on their own, kind of using the code. And then the one other piece that I'll show you at the very end of my talk is that we can also do what are called neutralization predictions. That is, once you estimate the parameters, you can try to then predict, knowing the parameters, how good is this antibody at shutting down spike protein or binding to it. So let me just show you some kind of validation of how the particle model does, and then I'll show you how we use it to make some predictions. And then that'll probably wrap us up in terms of time. So this is, again, this FD11A antibody that I've been using as my model all talk. And so here you see the three models, the monovalent classical model in red, that not very good bivalent model in purple, and then our particle model in black. So the first thing you see is we get the off rates really good, especially compared to the bivalent model, the bivalent ODE model. We don't get the on rates perfectly, but also there's a lot of variation in the on rates here, and we're getting them better than the ODE bivalent model gets them. We get the KD better because we get the off rate better, and we get two extra pieces of information: the second binding rate and the molecular reach. That is this distance over which things can interact. Okay. But this is only for one antibody. So a natural question is, well, what if you look at other antibodies? So we went and we did this for 5 different antibodies. And so here on the y-axis, you see the value the monovalent model predicts, the classical model. And on the x-axis, you see the value that we predict. And you see very good agreement for the on and off rates that the monovalent model predicts with what we predict. Okay, so we have good consistency with the older model for the things we think it's able to predict independently from our model. But we also get this extra information on the second binding rate and then the distance at which things can react. Then we did a second set of validation, which was to say, are the reach values that we're getting reasonable? And this is where Dan Nisley came in. So he's a postdoc in Charlotte Dean's lab, and he made we found 6 different of these antibodies that they actually had the all atom structures for. And he built coarse-grained molecular dynamics models of how those antibodies can bind to RBD. So here's the FD11A protein. Here's one receptor binding domain binding to one binding site on it, one arm, and here's another one. And then here's where they're tethered into what's supposed to be our approximation to the SPR attachment points. So here's where the RBDs are attached into space. And what we basically did, or what Dan really did, was he said, let's do an experiment where we think of the reach as the maximum distance between the tether points at which the whole thing can stay bound together. And so basically he fixed one of the tether points and he started pulling the other until it broke, until one of the RBDs popped off and it unbound. And so basically he defined in his model, the reach is the largest distance you can pull them apart before the bond breaks, basically. And so what you see up here is what he predicts from MD and what we predict from our SPR fitting to the SPR data. And you see quite good agreement.
I see.
So there's one antibody where the agreement is not amazing, but the rest of them, it's quite good agreement, I would say, between them. So that's kind of just another independent confirmation that these reach values we're predicting seem pretty reasonable. And I think I'll skip that one in the interest of time. Okay, so before I show you some predictions from fitting, let me just point out that there are problems or limitations with this model, just to kind of be fair and honest. So one problem is, of course, we're not fitting the model, we're fitting to the surrogate. So there's some accuracy being lost from doing that, fitting to this piecewise linear approximation. Another issue, though, is that you have to be a little careful about the experiments. So if you put too much antibody into the SPR device, we can't fit well. So here's an example where there was very large amounts of antibody you get much higher responses, and you see the fits are very poor. And we think what's going on there is when you have a lot of antibody, the antibodies can start binding to each other. And that's not in our model. So therefore, if it's not in the model, we're not going to be able to fit to that well, probably. Likewise, if you put too little antibody you flow over, then the system is going to be very noisy and you're not going to get much of a response. So you're not going to be able to fit well to that either.
Mm-hmm.
So you kind of have to have antibody, not too much, but not too little, just the right amount. And then for some antibodies, they just bind so strongly that they never come off. So here's an example where it comes off, barely comes off. If the curves look like this, there's not really enough features in them for a bivalent model to give you more information than the monovalent. So we would basically not expect that even though the bivalent model can fit them, that it's giving us more useful information than the monovalent model. So we also filter out antibodies that have this kind of response. And so, for example, Omer and his group went and they took 80 different antibodies from patients and they ran them over and they tried to do fitting, and 45 of them they found didn't have one of these issues. So a little more than half of them we could really apply our approach to. Some of them, this is just an inherent property of the antibody that it's hard to make it work in an SPR experiment because it just doesn't want to unbind. It binds too strongly. Okay. So in the last couple of minutes, let me just show you one prediction that comes out of this. So we can take all these antibodies, we can fit them, and we can get the parameters. Now, what can we do with those parameters? And so that's what I want to show you now. I want to show you two things. One, how the parameters relate to the ability of an antibody to neutralize spike protein or COVID, and also how we can do it predicting that ability. So this is an experiment, what's called the neutralization experiment, where they take actual virus with the spike protein on the surface and they incubate it with the antibody. The red curve is this FD11A antibody. The black curve is a version of it that only has one arm. And then they try to infect cells with it, and they see how well does it do at infecting cells when you've incubated it with different concentrations of antibody. Okay, so basically, and then they measure at some time what fraction of the cells are, roughly speaking, infected. Okay, so that's the neutralization experiment. And we can do an experiment to try to look at the efficacy of the antibody computationally, kind of in the following way. So we can pick an antigen concentration on the surface, and here we're doing a 2D model because in real life, the antigen, the RBD protein, is on the surface of the virus, which is a 2D surface. Okay, so here we're doing a 2D model for where the antigen lives. And so for each concentration of antigen, we can run a simulation for different antibody concentrations. And then at the same time that they looked at how many cells were infected, we can look at what fraction of the spike proteins have been bound by antibodies. And the specific statistic that people tend to look at is what's called the IC50. So it's for a given antibody it's for a given one of these curves, what's the antibody concentration where half of the antigen gets bound up? Okay, you can think of it as what's the amount of antibody where half of the spike protein gets shut down by being bound to it. And so you can measure that experimentally from these curves, and we can measure it experimentally in the model. And I'll come back to this in a minute. Okay, so before we look at that, let me just show you how the parameters that we estimate relate to the neutralization measures that they get out of these experiments. So here's the IC50 that they measure experimentally, and here's what we measure from the SPR experiments for the different parameters, just showing you the correlation between them. So this is just from the fitted parameters, how well do they correlate with this independent experiment? And you see very clearly the one that correlates the best is the reach. So the longer your reach is, the smaller the IC50, and that means you need less antibody to neutralize the antigen, i.e. the antibody is neutralizing the antigen better. Okay. And so this is the one that we see it's noticeably better than any other parameter, much better than the monovalent parameters. So kind of building from that, Anna, Omer's student, went and she looked at, and I'll tell you in a minute how we got this number, but she looked at how well does our bivalent model for specific antibodies predict the neutralization IC50 they measure experimentally. And so here's 6. I'm going to show you more in a minute, but this is kind of the warm-up that we did. And you see these 3, our model, which is the middle bar, agrees with the experiment exceptionally well. And the monovalent classical model doesn't agree well at all. This one, none of the models agree. We don't know what's going on there. And these two, our model agrees a lot less well. It predicts that the antibody is much more effective at neutralizing than it is, because bigger bars is worse neutralization, smaller bars is better neutralization. But there's an interesting thing. So basically, these three antibodies, which are able to neutralize, we're able to very well predict how well they're able to neutralize. And what Anna noticed that was very clever was that for all the ones where we were giving good predictions, the binding site was near the end of the RBD. So this is the RBD structure, a picture of it. And this is the binding site. And so here's the F13A, it's right at the end. The C121, it's right at the end. The FD11A is right at the end. And the reason that's useful is that the end here is actually where the RBD binds to receptors on your cell. So if your antibody's binding here, it's in some sense probably more likely to block the ability to bind to receptors on your cell than if your antibody is binding on the side. So taking that as motivation, this suggests that our method may be very good at predicting how effective an antibody is when the antibody's binding site is near this, what's called the ACE2 blocking site, near the tip of the receptor binding domain.
Let's see.
So we took the 25 antibodies from our panel that had a binding site close to the ACE2 receptor binding domain at the very end of it. And we looked for different antigen densities. What IC50, what neutralization do we predict versus what they measure experimentally? So here, y-axis is what we predict, x-axis is what they measure experimentally. And you see for very low densities, the correlation is not great. For very high densities, it's not great, but it's pretty good, or at least it's the best at this kind of intermediate antigen density, okay? And the interesting thing is, having done this, Anna went and kind of looked at the literature and said, okay, so here's the density that we find where we get the best prediction of the experiment. And that actually is consistent with what they estimate is actually the density of the RBD on the surface of the virus itself. So it suggests that if we use realistic antigen concentrations in our model, we actually can predict the ability of these antibodies to neutralize the antigen. So, okay, so that's just kind of one prediction, but one that we at least were very excited about. I know, sorry, I've gone a few minutes over, but I think I'll wrap up there. So just to summarize, this is kind of an integrated bivalent SPR modeling and experimental approach to estimate these binding kinetics and this kind of biophysical reach parameter. Our model is very consistent with the classical monovalent model for these parameters it can estimate, but it gives us this extra information. And in particular, this molecular reach, or this distance at which an antibody that's singly bound to an antigen can bind another antigen, seems to be the thing that best predicts neutralization and best correlates with the neutralization. So it suggests that the model may actually, at least in some contexts, give us a way to kind of, by running a panel of antibodies and calculating their kinetic rates and their reach, actually make predictions on which of them may be best at neutralizing the SARS-CoV-2 in this case. But we're looking at it now for other types of viruses in other contexts. So I think I'll stop there. Again, like I said, all the experimental work was done in Omer's lab, and Omer really, was a major contributor to every aspect of this, to developing the models and the methodology. Dan was a postdoc working with me on the models and methodology. Anna is a student with Omer that really did a lot of the using the models and the computational tools to do the fitting and then doing the analysis and kind of, I told you, like the hypothesis about the kind of where the binding site is. That was kind of her idea. Dan did these MD simulations. And as you can see, a ton of other people contributed in various ways. And I was funded by the NSF here in the US. And thank you all for coming in and attending the talk and for inviting me.
Thank you. Okay, so thanks so much, Sam. It's time for the Q&A. So yeah, please feel free to unmute yourself and have a question.